March 25. Benedict solution is used to test for reducing sugars. Glucose is a reducing sugar. A student estimated the concentration of glucose in a solution. I have an unknown solution. And I want to test. I want to know its concentration. So what am I going to do? I will prepare test tubes with concentrations that I know. يبقى he will tell me the method بقى. How did he know this solution? The student uses this method. Label 6 test tubes, 0, 0.5, 1, 1.5, 2, and then this is the unknown one. Complete the table by writing the volumes of the glucose solution and the distilled water that you used. These were not here. You need to calculate them. طيب. For some people, they will think, okay, it's going in sequence 0, 1, 2, 3, 4, 4, 3, 2, 1, 0. This is how it's going. But I still need to know the rule. I still need to know the rule because he might get a question without a table. In order to calculate the final concentration of any solution, in order to calculate the final concentration for any solution, how do I do it? You will write the volume you took from the glucose solution, volume of glucose used, over total volume, which is glucose plus water, times the concentration you took from, for this question, he used the 2% solution. This will give me the final concentration. طيب, what is the final concentration? 1.5. How much did I use from the glucose solution? I don't know. What is the total volume? All of them are 4. 4, 0, 3, 1 is 4, 2, 2 is 4, 0, 4 is 4. So this is probably also 4. What is the concentration that I already took from? 2%. Can I get this? Yes, I can. I'll put this here times 4 and then divided 2. It will give me the number. Okay? Divided 2. So we agreed that this will be 3 and this is 1. Use a syringe to put the volumes of the glucose solution into the test tubes. Into these test tubes, I will put these volumes into the test tubes. Use the same syringe to put 4 cubic centimeter of the unknown in test tube U. I took 4 cubic centimeter from this unknown with the same syringe. Come on, this is a source of error. I use this syringe to put all these, and then I use the same syringe to put solution U. Use a clean syringe to put the volumes of distilled water. I will put the water بقى in the test tubes. Put the test tubes in hot water bath, 80 degrees. Sorry, I missed a point. Clean syringe to put 4 cubic centimeter of Benedict in all. So this is a constant variable. Also, the total volume of the solutions is constant. The total is constant. Put it in a hot water bath, 80 degrees, start the stop clock. This is the water bath for the Benedict. Wait for 5 minutes, remove them all, record the color of the liquid. This is a source of error. It is difficult to judge color by eyes. Okay, these are the results. Prepare a table. Changed variable is on the x-axis, results on the y-axis. This is my changed variable. This is my results. I changed the concentration of glucose. Please, the units are in the headlines only, no units in the table. And this is the unknown. Observation slash color. Okay, I recorded the colors. Tab now, can I know the concentration of U? Yes. It gave me orange yellow between these two colors. So the concentration is between these two. What was the range he gave you? From 1.1 to 1.4, because it was between these two. If it was 1, it will be yellow. If it is 1.5, it will be orange. No, it is not this or that. It is yellow-orange. So I can write any concentration between them. Okay? Okay. State two variables that were kept constant. I already marked some of the stuff. El volume of the Benedict, el final volume of the solution, anything that will affect the results. Identify the possible source of error in steps 2 and 3. Steps 2 and 3, what was the error? Using the same syringe. Using the same syringe, what is the error? It will cause contamination. It will mix these with the unknown solution that I have. طب what is the other part of the question بقى? State the effect of this error on the results. What is the effect on the result? The effect will be, what is the final effect? When you use the same syringe, you increase the glucose for you, so the concentration of unknown will be higher than it should be. It will be higher. You have to write increase. He wants to know the effect. Okay. Eggs contain protein called albumin. What does the albumin do? It turns from cloudy to clear when it is digested by pepsin. Plan an investigation to determine the effect of A. Effect of pH. So this is my independent variable on the digestion of albumin by protease. I will write my steps. Number 1, what am I going to use? I'm going to use test tubes to put the albumin in. 5 test tubes. Each of them with a different pH. How do I change pHs? Using something called buffer solution. يبقى 5 test tubes, different pHs using buffer solution. And then same. What am I going to keep the same? This is an enzyme question, and you change the pH. The rest, the temperature, the volume of enzyme, the volume of pepsin, the volume of albumin, the concentration of albumin, everything should be the same. These are my control variables. What am I going to measure the results? He always give you a hint. He said that if pepsin is working, it will change the albumin from cloudy to clear. So I will record the time taken for the solution to become clear. Improvement, safety precaution, repeat, improvement, it is maintain the temperature so it doesn't change using thermostatically controlled water bath. Wear goggles, repeat the experiment. I have to write at least 3 times and calculate an average. 6 marks. طيب. Describe how to do emulsion test. Emulsion test. I will add ethanol on the fat solution and then water and then shake. This is 1 mark and this is the second mark. Okay. Question 2. Draw a diagram. Draw a large diagram of the whole oak leaf. The usual, we draw the outline. I have to draw it like it looks the same with these parts, wavy outline, okay? And a small stalk, midrib, double lines, veins, 1, 2, 3, 1, 2, 3. This is a bit branching. If it's a bit branching, you can add it. A big drawing that looks the same with the same proportions. Clear outline. The upper lobes are at least 3 or 4. 1, 2, 3, 4. Here 1, 2, 3, 4. Up and down. After the drawing, there is the magnification question. Line PQ represents the width of the oak leaf. Okay? Measure line PQ using your ruler. It will be in centimeter. I will multiply by 10 to change it to millimeter. 58. Calculate the actual width. Image over magnification. Image over magnification, which is 1.4. Okay, 58 over 1.4. And then my answer will be rounded to two significant figures. Okay. Figure 2.2 shows photographs of the oak leaf and the hollyhock leaf. He gave me two pictures. He wants you to compare. For example, the first point he gave it to you. This one is longer than its width, while this is wide. The length and the width are similar. But what else? I can say that this have indentations. Indentations. There are parts that's going inside, while this one has no indentations. This one has smooth outline, while this one has a serrated or a zigzag edge. I can talk about the distribution of the veins. The veins here all start from a single point. Okay? While here, veins branch from a single midrib. Take care, he told you do not reference to the size. So I cannot say one is bigger than the other. So these are the differences. We said this one has large indentations, this does not. This is smooth, this is serrated, or I can talk about the pattern of the veins. Question B, investigated the effect of light intensity on the surface area of the leaves. I am going to change. This is the independent and this is the dependent variable. I'm going to change the light intensity and measure the surface area. 100 soya beans were planted, put in the shade. 100 were put into full sun. The soya beans were allowed to germinate and grow for 30 days. After 30 days, the oldest leaves and 3 of the youngest leaves were removed. Surface area was measured. Right. What is my dependent variable? Dependent. No. I was measuring the surface area. This is my result. Why the students use large number? I can write two things: to get a representative sample, to make sure that the sample I got represents the actual population, and to avoid bias. I do not want to be choosing certain plants. I just take a big sample randomly to avoid bias. Okay. The student estimated the surface area using this method. How do I do this method? I draw the outline of the leaf on the graph paper, and then I remove the leaf. Count the squares that are fully covered by the leaf. But what about the squares on the edge? If it is more than half covered, I include it. Include the squares that are more than half covered. Each of these points had one mark. I need two. بس يعني that makes sense. Yeah. This question had a very small كده trick. يعني not a trick, but a bit of a change. The results of the investigation was shown on the grid using the data. Plot a bar chart. Usually, before this exam, he used to tell you plot a bar chart of the lighting condition against the surface area. So you would choose two columns and you would ignore this one. He did not say this here. He said plot all this in the bar chart. What am I going to do? The left is on the x-axis. The results on the y-axis. I'm going to label the y-axis mean surface area. Mean surface area of leaves. And on the x-axis, I do not have to label it. Or I can write both light and age. Light condition and age. Because I will put both. I put my scale. The biggest number is 78 and I have 8 boxes. So I put it 0, 10, 20, 30, 40, till 80. I drew my bars. The bars are equal. They have to be wide to take a big space. Spaces are equal. How am I going to call my bars بقى? Both names. Full sun and young. Full sun and young. Shade and young. Full sun and old, shade and old. Full sun and old, shade and old. He wants both. Okay? And then a percentage increase question. The students calculated that there was a 15.6 increase in the size of the young leaves. Calculate the increase in the surface area of the old leaves. Full sun and shade. Okay. Full sun and old. Shade is 58. How do I calculate the percentage increase? This is the final, this is the increased one, and this is the original. Final minus original over original times 100. 78 minus 58 over 58 times 100. This is how I calculate my percentage increase. 1 decimal place. So what are two conclusions? Look at this. I can see that old leaves have a larger surface area than the young leaves. Both old have a larger surface area. Also, the leaves in the full sun have a larger surface area than the leaves in the shade. These are the two conclusions. Old leaves are larger than young. Full sun leaves are larger than the ones in the shade. The last question is also about the hydrogen carbonate. Aquatic plant was placed in red hydrogen carbonate under bright light. It will photosynthesize. What is the final color of hydrogen carbonate? It will make photosynthesis, so it will absorb the CO2. The CO2 will decrease in the solution, يبقى the color will be purple. This is the end of the exam.